Think of your immune system like a super loyal guard dog. This dog is incredibly powerful and really good at sniffing out and attacking anything that doesn't belong. It's constantly on patrol, wiping out tons of viruses, bacteria, and other tiny threats. We wouldn't last long without it, but there's a catch. Those sharp teeth could easily hurt the family it's supposed to protect, not just the enemy. The biggest puzzle about our internal defenses, our immunity, isn't how they attack, but how they know when to back off. For decades, scientists thought they had the answer. They figured the immune system's basic training happened once. This early education, which goes down in a small organ called the thymus, teaches the immune system's fighter cells, called T cells, to recognize the body's own stuff as self. This is called central tolerance. But this wasn't the complete story. It couldn't explain how the immune system stays so well behaved throughout a whole lifetime, long after its first schooling is done. This year, the Nobel Prize in Physiology or Medicine honors the scientists who solved this puzzle. The award goes to Mary E. Brunkow, Fred Ramsdell, and Shimon Sakaguchi for their discoveries concerning peripheral immune tolerance. This system is a lifelong peacekeeping operation that constantly keeps the guard dog in check. It is the reason most of us are not consumed by our own defenses. This is the story of how those peacekeepers were found, how a master genetic switch that controls them was identified, and how this knowledge is now being used to rewrite treatments for autoimmune diseases, make organ transplants safer, and sharpen the fight against cancer. By the end, you will understand why this discovery was deemed worthy of medicine's highest honor in 2025. The idea that the immune system could be taught restraint is not new. In 1960, the Nobel Prize in Medicine was awarded to Sir Frank McFarlane Burnett and Sir Peter Medawar for establishing the concept of immunological tolerance. They showed that our immune system figures out what's ours and what's not while we're growing up. In essence, they discovered the principle of central tolerance. If you introduce a foreign substance to an animal while it is still in the womb or just after birth, its immune system will treat that substance as self for the rest of its life. This discovery created a wave of wonder and optimism. It explained why we don't normally attack our own organs, and opened the dream of one day tricking the body into accepting a transplanted organ from another person. The process works through a rigorous screening program in the thymus, a small gland behind the breastbone. As new T cells mature there, they are tested. Any T cell that reacts too strongly to the body's own proteins is ordered to self-destruct. Only those that pass the test are allowed to graduate and enter circulation. But a nagging puzzle remained. This central schooling, however effective, was not perfect. Some self-reactive T cells always slip through the net and escape into the body. Furthermore, our bodies are not static. We get injured, we get infections that can look a little like our own cells, and our tissues change as we age. Why don't these events constantly trigger autoimmune diseases in adults? Scientists suspected there had to be another mechanism, a second layer of control operating out in the periphery, that kept these escaped warrior cells in check. For a time, some researchers pursued a theory of suppressor T cells, cells that might actively calm down immune responses. But the evidence was weak, and some experiments were shown to be flawed. The entire field fell into disrepute, becoming something of a scientific backwater. The idea of active suppression was mostly abandoned. It would take a scientist willing to swim against the current to prove that the ghost of a peacekeeping cell was, in fact, real. The solution to the puzzle of peripheral tolerance was pieced together over nearly a decade through the work of 3 scientists who started from completely different places. One was an immunologist chasing a function, the other 2 were geneticists chasing a disease. Their paths would converge to provide an answer so complete and elegant that it would reshape the field. The first piece of the puzzle came in 1995 from Shimon Sakaguchi, then at the Aichi Cancer Center Research Institute in Japan. He was intrigued by a strange observation from years earlier. Scientists knew that removing the thymus from a newborn mouse weakened its immune system. But if the surgery was done on a mouse that was just 3 days old, something opposite and violent happened. The mouse's immune system went into overdrive, attacking its own body and causing a storm of autoimmune diseases. This suggested that the thymus of a slightly older mouse was producing not just warrior cells, but also some kind of protective cell. Dr. Sakaguchi was convinced that a population of security guards must exist. He spent more than a decade trying to find a way to distinguish these hypothetical peacekeepers from the aggressive warrior T cells. Finally, he found a marker. He discovered that a small fraction of a type of T cell known as a CD4 helper T cell also carried a protein on its surface called CD25. His experiment, published in the Journal of Immunology, was decisive. He took mice that had no T cells of their own. If he gave them a population of CD4 T cells from which he had removed all the cells marked with CD25, the mice developed severe autoimmune diseases. Their immune systems, lacking any restraint, attacked their own tissues. But if he added back just that small fraction of CD25 positive cells, the mice remained perfectly healthy. He had found them. He had isolated the immune system's peacekeepers, which he named regulatory T cells, or Tregs. The discovery was met with considerable skepticism. The field had been burned before by the flimsy evidence for suppressor cells, and many immunologists wanted more definitive proof. That proof would come from an entirely unexpected direction. In Bothell, Washington, at a biotech company called Cell Tech Chiroscience, 2 researchers, Mary E. Brunkow and Fred Ramsdell, were working on a different mystery. Theirs began with a peculiar strain of mice called scurfy. These mice had first appeared as a spontaneous mutation in a laboratory in Oak Ridge, Tennessee, during the era of the Manhattan Project. Male scurfy mice were a picture of immune collapse. They were small, had scaly skin, and their organs were massively swollen with out-of-control immune cells. They died within a few weeks of birth. Doctors Brunkow and Ramsdell reasoned that if they could find the single gene responsible for this catastrophic failure of immune control, it might offer a clue to what causes autoimmune diseases in humans. The task was enormous. In the 1990s, finding a single mutated gene on the massive X chromosome, where the scurfy mutation was known to reside, was like searching for a specific grain of sand on a vast beach. After years of painstaking genetic mapping, they narrowed the location down to a region containing 20 possible genes. They examined each one, comparing the DNA of healthy mice to that of scurfy mice. With the 20th and final gene, they found it. It was a gene no one had seen before. They named it FOXP3. Their work did not stop there. They suspected that a rare and brutal human disease that affects young boys might be the equivalent of the scurfy mouse. The disease was called IPEX syndrome for immune dysregulation, polyendochropathy, enteropathy, X-linked. Working with pediatricians, they obtained DNA samples from boys with IPEX. Their hunch was correct. The boys all had mutations in the human version of the FOXP3 gene. In 2001, they published their findings in Nature Genetics, showing that FOXP3 was the cause of both the scurfy condition in mice and IPEX syndrome in humans. The final piece of the puzzle clicked into place in 2003. Shimon Sakaguchi's lab, along with others, published a paper in Science demonstrating that FOXP3 was the master switch for regulatory T cells. The protein made from the FOXP3 gene is a special kind of protein called a transcription factor. Its job is to turn other genes on or off. The work showed that the FOXP3 program is what instructs a normal T cell to become a Treg. The convergence was complete. Dr. Sakaguchi had found the peacekeeping cells. Doctors Brunkow and Ramsdell had found the genetic program that creates them. The existence of a dedicated system for peripheral tolerance was no longer in doubt. The Nobel Committee noted that these findings together laid the foundation for an entire new field of research by explaining how the immune system is kept in check outside the thymus. The discovery of regulatory T cells and their master switch, FOXP3, was like finding the user's manual for a complex piece of machinery. For the first time, scientists could begin to understand not just that the immune system restrained itself, but how. The Tregs, programmed by FOXP3, are the career peacekeepers. Their job is to intervene in heated situations before real harm is done. Then there are the effector T cells, the soldiers of the immune system, which are eager to attack but can be calmed down. Finally, there are antigen-presenting cells, or APCs. These are the intelligence officers that survey the body for signs of trouble and present evidence in the form of protein fragments called antigens to the T cell soldiers. Tregs use at least 4 main strategies to maintain peace. First, they soak up essential supplies. To multiply and mount a full-scale attack, effector T cells need a chemical growth signal called interleukin-2, or IL-2. Tregs are covered in high numbers of receptors for IL-2, far more than other T cells. They act like sponges, absorbing the available IL-2 in an area. Second, they release calming chemical messages. Tregs produce their own set of signals, called cytokines, that have the opposite effect of the immune system's alarm bells. Molecules like IL-10 and transforming growth factor beta, TGF, spread through the local tissue, essentially broadcasting a message to other immune cells to stand down and that the situation is under control. Third, they directly influence the intelligence officers. Tregs can interact with APCs and persuade them to change their tune. Instead of presenting an antigen as a 5-alarm fire, a Treg can induce an APC to present it in a calming, non-threatening way. This re-educates the local T cells, teaching them that this particular signal is a false alarm. Fourth, they apply the brakes directly. T cells have a system of accelerators and brakes on their surface to control their responses. These are often called immune checkpoints. One of the most important brake pedals is a protein called CTLA4. Tregs have high levels of CTLA4 on their surface. By physically interacting with other immune cells, they can use CTLA4 to deliver a powerful stop signal, preventing an attack from getting started. This particular mechanism connects the science of tolerance to a recent revolution in cancer treatment. The 2018 Nobel Prize was awarded for the discovery of cancer therapies that work by blocking these checkpoints, effectively cutting the brake lines to unleash the immune system against tumors. The work of this year's laureates explains why those brakes exist in the first place. They are a primary tool used by Tregs to maintain peace. The Nobel Assembly often recognizes discoveries decades after they were made, waiting until their full importance becomes clear. The decision to award the 2025 prize to Drs. Brunkow, Ramsdell, and Sakaguchi reflect the fact that their work has now completed the full journey from a fundamental biological question to a new frontier of medicine. So why is 2025 the year for this award? It's now that fundamental science is a settled part of immunology textbooks. The genetic link to human disease is undeniable. Most importantly, therapies based directly on this knowledge are now moving through clinical trials. Researchers are developing treatments that boost Tregs to fight autoimmune disease and prevent transplant rejection. The inverse logic disrupting Tregs to fight cancer has the potential to become a pillar of oncology. The field has crossed a crucial threshold, moving from experiments in mice to the complex reality of human therapy. The timing of this Nobel Prize acknowledges that the seed planted 3 decades ago has now grown into a tree that is beginning to bear fruit for patients. The knowledge of how peripheral tolerance works has provided a blueprint for a new generation of medicines. One approach involves gently nudging the body's existing peacekeepers to multiply. As we've seen, Tregs are highly sensitive to the growth signal IL-2. While high doses of IL-2 are a powerful stimulant for the entire immune system, researchers have found that administering very low doses can selectively encourage Tregs to expand without revving up the aggressive effector cells. This strategy of low-dose IL-2 therapy is being tested in clinical trials for a wide range of autoimmune diseases, including lupus, rheumatoid arthritis, and inflammatory bowel disease. The key is finding the right dose, enough to boost the peacekeepers without waking the warriors. A more direct strategy is known as adoptive Treg therapy. This involves taking a sample of a patient's own blood, isolating the rare regulatory T cells, and growing them to massive numbers in the lab. These expanded autologous Tregs are then infused back into the patient, flooding the system with a fresh army of peacekeepers. The most advanced approach involves engineering Tregs to be even more specific. This would be a major advance over the lifelong immunosuppressant drugs that transplant recipients currently rely on. Similar programs are being developed for autoimmune diseases like type 1 diabetes and inflammatory bowel disease. The discoveries that won this year's Nobel Prize have given scientists a user's manual for the body's peacekeeping forces. The next chapters of medicine will be about learning to write new instructions for them, moving from managing a delicate balance to achieving personalized control over the immune system. In the near term, over the next 5 years, we can expect to see the first fruits of this research reach patients. Clinical trials will produce more detailed playbooks for using low-dose IL-2, refining the dosing schedules to maximize the expansion of Tregs while minimizing side effects. We'll probably also see the first regulatory approvals for adoptive Treg therapies for specific conditions, like preventing organ rejection in transplant patients. As that happens, the manufacturing will get faster, cheaper, and more reliable, making these complex living drugs more accessible. But over medium term, scientists are working on combining Treg therapies with other technologies. One promising area is the use of biomaterials. Imagine a small biodegradable scaffold or gel that can be implanted directly into an inflamed organ, like the pancreas in type 1 diabetes or the joints in rheumatoid arthritis. This material could be loaded with factors that attract and sustain Tregs, creating a localized hub of immune suppression exactly where it is needed, without affecting the rest of the body. Alongside new therapies, we will see the development of smarter diagnostics. Blood tests that can accurately measure the number and functional strength of a person's Treg population could allow doctors to monitor their tolerance health, and perhaps even predict an autoimmune flare before it begins. In the long term, the goal is to achieve truly personalized immune tuning. A day may come when a doctor can dial a patient's immune response up or down with precision, targeting a specific organ or even a single protein. In cancer, the future lies in greater precision as well. Rather than systemically blocking immune checkpoints, which can cause autoimmune side effects, future therapies may be able to selectively identify and disable only the Tregs that have infiltrated a tumor. This would unmask the cancer to the immune system while leaving the body's vital network of peripheral tolerance fully intact. The work of Mary Brunkow, Fred Ramsdell, and Shimon Sakaguchi did more than solve a long-standing puzzle. It gave us a new way of thinking about the immune system. The medicine of the future will be built on that understanding, learning not just how to fight our body's enemies, but how to foster peace within ourselves.